All right. Welcome, everyone. It is my absolute pleasure to introduce tonight's speaker, Dr. Meredith Zettlemoyer. Um, Meredith is an assistant professor at the University of Montana. After studying biology and English at the University of Virginia, she went on to earn her PhD in ecology, evolutionary biology, and behavior at Michigan State University, where she studied extinction of tallgrass prairie plants. Meredith's postdoctoral work at the University of Georgia focused on how climate change affects alpine cushion plants. Meredith joined the University of Montana in 2023, where her lab studies how plant populations, communities, and plant pollinator interactions respond to global changes like advancing snowmelt and more frequent wildflower fires, excuse me. They also study the reproductive ecology, pollination, and trait variation in Missoula's endemic Phlox species. Outside of research, Meredith enjoys hiking, playing with her dog, and musical theater. Thank you so much, Meredith, for joining us. We're so honored to have you tonight.
Thanks, Alex. Um, yeah, I am really excited to share with you all some of my lab's very, very new work on Missoula phlox. So I was kind of already did this, but I thought I'd start with, oh, my slides don't want to move. A little bit of background. I'm a military brat, so I've lived all over the US. The most boring place was Nebraska. The most exciting place was Oahu. So hi to the person from Hawaii. That's cool. Um, After moving around a lot as a kid, I went to the University of Virginia for my undergrad. Yeah, as Alex said, studied biology and English, but did a stint with AmeriCorps in a state park, which made me super interested in ecology. And so I joined this lab working on life history, so things like timing of germination, timing of flowering, in Campanula americana, or American bellflower, this lovely guy down here. Got super interested in sort of responses to global change. So I moved to Michigan to then do my PhD working in tallgrass prairies, which was several years of working in grasslands with grasses taller than I was. It was very, very itchy field work, but we learned a lot about what causes a lot of these prairie plants to totally disappear from the landscape. After that, I moved to the University of Georgia, where I did my postdoc, but that's a little bit misleading because we actually did all of our field work in the Front Range in Colorado, right outside of Boulder at the Niwot Ridge LTER. So that's where I started working on alpine cushion plants, and so I'll explain what cushion plants are in a slide or two. But really thinking about how earlier snowmelt kind of messes with their interactions with pollinators, their interactions with each other, and then how well they survive. So then in 2023, I showed up in Missoula. And so my lab is still working in Colorado on these three cushion species. We're also doing a lot of work on how forest understory communities respond to shorter and shorter intervals between wildfires, largely in the Sapphire Mountains. We've started some work thinking about how a lot of different types of global changes can affect plant pollinator interactions. Um, and then our most local system, the one I'll tell you about today, is we're working with this really cool cushion plant community that we find in Missoula, with a particular focus on the endemic Missoula phlox. Okay, so cushion plants are near and dear to my heart, obviously, but they are this growth form of plant that looks, I mean, they literally look like a little pillow. So they're very compact. As you can kind of see in this image, everything is very much sort of rounded, and they grow really low to the ground, typically only a couple inches off. And that's because cushion plants are normally found in these really harsh environments above tree line. So in places like alpine tundra, like you can see here. And so, in these habitats where there's really harsh winds, so being low to the ground is going to be really beneficial. You want to be low to the ground also to get insulated under snowpacks. You're not exposed to, freezing air temperatures, especially in the spring. And so, this is a growth form that's super well adapted to these high elevation environments. But what's really cool is that there is a cushion plant community that's found at pretty low elevation in Missoula on these kind of arid, very windy ridges that we see across the parks and preserves in the city. And so these cushion plants include obviously Phlox missoulaensis, the local endemic species that I'll tell you a little bit more about, as well as species like Douglasia montana. So for those of you in Missoula, this is going to be probably the first thing you see in April. It's the earliest flowering species, at least on some of the local parks. Species like cutleaf daisy, this Erigeron compositus, um, cushion buckwheat, this Eriogonum. There are a handful of others that I pin picture here. There's like 6 or 7 species that make up this community. But there are these species that, based on their growth form, we'd normally find in like Glacier National Park or the very top of like the Beartooth Mountains, but they're here instead. So Missoula phlox is special because it's actually a cushion plant that is only found on these arid, windy ridges in the North Hills in the Missoula Valley. So some of the known populations are marked in red on this map here. So this species has a really restricted range for known sites, but where it is found, fortunately, it is really well abundant. So if you go out to, for example, Waterworks Hill in about May and early June, it'll be blanketed in white, and that's mostly the phlox. But the thing here is that these preserves have trails through them. They're trails that are very popular. They have beautiful mountain views, as you can also see in this photo. And so the habitat that these species survive in is pretty threatened, mostly by off-trail human and animal, particularly dog walking use, and some trampling to the plant. So very healthy populations where they're found, but restricted range in a habitat that's fairly threatened. And so in 2025, Phlox missoulaensis was listed as a state species of concern, kind of based on the fact that it's very rare, only found in the North Hills. It's highly threatened. As I mentioned, it's just, in a very trafficked area. Um, the good news is that where it is found, it is stable population. So we can see this kind of on this green end of the graph here. But this does mean that in the state, it's ranked as an S1 species or a species of pretty severe concern for conservation. And the state rank reasoning also highlights a need for a lot of ongoing monitoring, as well as continued implementation of all these different land conservation practices that could help out the flocks. And so my lab, we started here in 2023. This was one of the first plants I noticed my first summer here because it was everywhere in early spring, and showed a lot of variation. It was in these really cool habitats where I didn't expect them to be. And so this is a system that we're excited about because we've just started working on it. The caveat I'll give you today is I'm going to run through these 5 very different sounding stories. And that's because we're kind of like, we're picking at the system right now to learn what we can. Um, so we don't have complete stories for most things. We're kind of being pointed in the direction of interesting questions, but everything I'm going to be showing you today is pretty early data. Okay, so we're going to start just thinking about sort of the taxonomy of these species. How do we tell it apart from some of its close neighbors, and what are some of the potential patterns of relatedness with other phloxes? So, Phlox missoulaensis has kind of a messy history, actually. So, historically, it's generally been considered a variety of 2 other species, Phlox kelsey and Phlox pulvinata. So, I'll give you a little bit of an overview of each of these guys. But Phlox kelsey is a species that is generally found from kind of the southern end of Idaho, over here. And then kind of into this is Missoula Valley and then a little bit farther north. And then also tails off a little bit into Wyoming. This is a species has a pretty broad environmental tolerance. So it's considered to have 3 different varieties. So variety Kelsey has been found in these seasonally wet alkaline or high pH soil meadows. Var salina is typically found in very alkaline, so you can think of this as kind of like salty soils. And then historically, Var missoulaensis was considered to be this variety that was endemic to Montana, particularly to the Missoula Valley, and only found on the words in the like herbarium specimens was wind blasted mountain ridges. So very dramatic. And this is a species that, it kind of looks a little bit succulent. The leaves are kind of shiny, they're kind of thick looking. The other species that it's often sort of labeled a variety of, although not the only ones, is Phlox pulvinata, and that's cushion phlox. So this is the species that we would typically say has this very traditional cushion plant form, compact, low to the ground, looks like a very comfortable pillow if you don't actually touch the leaves. Um, and it's going to be what we would normally find in subalpine and alpine areas, so those typical high elevation habitats. And then pulvinata is found a bit more broadly across the western US. So Phlox newzealensis for a long time, if you like look at herbarium specimens, was considered a variety of one of these two species. But in 1944, Thomas Wary basically said, based on this 1932 collection that was made in Missoula, that there were some distinctions about the species that would maybe make it its own. So he cited these longer and wider leaves as well as longer sepals, which are kind of the green leaves that like sort of cup the petals of a plant, relative to pulvinata. It had kind of an intermediate growth form, so it's definitely more cushiony than Kelsey. I've already showed you a couple of photos where it looks like very much like a little puffball pillow. But it can also be a little bit sprawly or decumbent, so it can kind of break that cushion plant form a bit. It's considered to have narrower leaves and be less succulent than Kelsey, so not have quite that like thick, shiny, I'm going to hold a lot of water look. And then it also had pretty hairy or pubescent leaves. So he's like, these traits kind of make us think like it could be its own thing. Um, and then over time, it's been increasingly recognized as its own species. And that kind of came to fruition in the 2025 meeting where they were like, okay, this is a state species of concern. We're super interested in it. We're going to consider its own thing for at least conservation purposes. So one of the things that my lab is really interested in doing, and here we're starting to get into kind of planned work, is we really want to try to figure out what are some of those morphological or trait-based delineations between these three species. So an undergrad in my lab next semester is going to be working with herbarium specimens, such as this image here. And on those, we can measure some of these key traits that Wary listed and that are in like sort of the keys. So things like leaf width and length. We can look at individual leaves, like on this red line that you probably can't see too much of what it's outlining, but we can put that into imaging software and estimate leaf width and length. And then this yellow line here, we can find the sepals that are associated with an individual flower and measure how long those are. We can look at the herbarium specimen label and get the data for things like flowering time. And what we're hoping to see is that if we were to sort of map these species in terms of their traits, we'd end up with an axis that is leaf traits, an axis that's maybe sepal length. These axes could change depending on what ends up being the most important. But hopefully what we'll see is that, there might be a lot of variation, there might be some overlap, but hopefully, Kelsey has some set of traits, succulent, short leaves. Pulvinata has some traits, maybe shorter leaves, cushion plant growth form, maybe Missoulaensis is somewhere in the middle, but hopefully we'll be able to kind of sort out what those key differences are in a more quantitative way. So there are also some cool things about Phlox and its spatial distribution that are making us really curious about how it's speciating or how it's become the process of becoming its own species, if it is. So the first thing, as I've already mentioned, is that Phlox missoulaensis is spatially isolated to the Missoula Valley. So these are the ballpark areas where some of it's been found. Some of these are the specimens with varieties on them, whereas these other two guys are a bit more widespread. Um, This is more of a fun fact. It's not really too much related to the gene flow. But something really strange about it is that phlox is also only found below the glacial Lake Missoula high water line. So glacial Lake Missoula covered Missoula Valley. So this map here showing the extent of the lake, this point right here in orange is the Montana Natural History Center. So all of this would have been totally below water about 12,000 years ago. And um, what was happening was the Cordilleran ice sheet was basically damming the Clark Fork River. The rising water behind this basically like dam of ice weakened the glacier until the water burst through, and this flood that kind of sped through to the Pacific through Oregon and Washington. But over time, glacial Lake Missoula filled in and emptied, refilled in and emptied. And so we ended up with these. This is where you can see these strand lines in this photo of Mount Jumbo, where the high water line was at various points in time. And so something that's just kind of funky is that even though there are cushion plant communities, so that the Douglasia, the cushion buckwheat, etc., at the very top of Mount Jumbo, you actually cannot find phlox there. And I couldn't tell you like what about being below the high water line makes that habitat suitable at this point in time. But a little bit of an aside. So the other thing about the phlox is that it's also what we would say temporally isolated. So Phlox missoulaensis flowers. So this is frequency of flowers on this y-axis, and then just month on the x. And so Phlox missoulaensis flowers in April to May. Kelsey kind of peaks in June, and then pulvinata peaks in July, which makes sense for being a high alpine species where the growing season is a little later. But what this means is that because these guys are flowering at very different times, they're not likely to like have pollinators flying between their populations and cross-pollinating, kind of sharing those genes. And so they're not only spatially isolated, but they're temporally isolated. And so then you're not getting a lot of interaction with each other. And so this leads this spatial and temporal isolation is leading us to ask a couple questions about, what is Phlox missoulaensis' actual closest sister species? So who is its closest relative? Is it the Kelsey? Is it the pulvinata? We have a couple other candidate species that we think it could be. Um, is it still very closely related to that sister species, or is it, fully separated from it? Can we truly say that it's its own thing at this point in time? And to answer that, we need to first like build a phylogeny, which is basically I don't have an image of it on this slide, but a thing that kind of looks like a tree where branches that are very close together mean species are super closely related, and then species that are very far apart would be less closely related. But to look at things like gene flow, we want to take this approach very similar to an approach that's been taken for Phlox pungens, which is an endemic found in Wyoming. So in this species, I have to actually count these populations, my gosh. They know of 10 populations across these two river basins, the Wind River Basin and the Green River Basin. And this plot is called a structure plot, and it's showing the mixture of genes from different populations. So in the Wind River Basin, at least these four populations, Beaver Rim, Sheep Mountain, Cedar Ridge, and Red Canyon Rim, because they're all the same color, that means that they're all interacting with each other. So all the genes that are being passed along because of pollination and like interactions between the species is within those four populations. In the Green River Basin, it's almost all blue. So they're mostly getting gene flow from each other. There's a little bit of orange, so there's a little bit of gene flow happening, but not much. And then there's this one population in purple, this red butte, that's totally different. And so we can look at the relationships between the populations of Phlox missoulaensis, as well as between Phlox and these sister species, and say something about, are these guys still sharing genes in some way? So are they still able to kind of interbreed? And so maybe speciation isn't totally done yet. And so to address that, we're going to be collaborating with the Leela Fishman's lab at the University of Montana to do some genomic work. So we're collecting tissue we've got three of the four known populations of Missoulensis in the North Hills. So that's this pink dot here in Missoula. We have a wonky population in Helena National Forest right here that is Kelsey var missoulensis, but the habitat looks like what we see in Missoula. So it might be like a weird disjunct intermediate population. We're not sure. And then we have one population each of Kelsey pulvinata, and then two other species, Phlox multiflora and longifolia, from Montana. And then we also have a population of Phlox condensata, which is a high alpine species found in tundra in Colorado. And so um, shout out to Minody Asher. She's been leading this work wonderfully for the last year or so. But between this field tissue and then getting some tissue from herbarium specimens of these species, as well as specimens that have kind of mixed up annotation histories. So things that, maybe were once called Kelsey and then 20 years later, someone was like, oh no, this is Missoulensis actually. So the ones that are a little bit confusing, and maybe some representative other Western phloxes, we're hoping to use that to build a new phylogeny, and look at the gene flow between the populations and these species and really try to kind of resolve the evolutionary relationships or, who is most closely related in this system. Okay. So Phlox, as I kind of just outlined, is super morphologically challenging. And the overlap in its traits with things like Kelsey and pulvinata is not just an issue across its potential sister species, but also is actually inherent to kind of the massive amounts of trait variation that we see within Missoula phlox itself. And so what I mean by that is that there's a huge bunch of different ways that this species can look. And so it can be extremely variable in its size. So this is a plant that was like probably the size of like two watermelons. So a humongous plant, very sort of isolated from areas with foot traffic. And then we have other plants on the right side where this plant is probably only like 10 little rosettes wide. So it's probably like the size of, well, I'd maybe say two quarters. There's also a ton of variation in its growth form, as I alluded to earlier. So it can be this very compact, very pillowy, very traditional looking cushion plant, or it can be super, super sprawly. So this image on the right I tried to outline kind of the edge of the greenery for the phlox leaves. And so you can see that it doesn't look like a nice little pillow. It's very much sprawling across the space that it can. Can have a lot of variation in shape. So typically phloxes are five-petaled, but there's a bunch of individuals that can have six or seven petals. That's actually because of a single mutation that's well known across the phlox genus. But what's been interesting to us is the variation in the petals. So they range from being very narrow and long, such as this middle photo, two on the right being these very like fat wide petals. And so these fat wide petals look to us like, for a bee that was trying to pollinate, that looks like a beautiful open landing pad that would be really easily like sat on to collect pollen and nectar. And then the last trait that I want to sort of highlight is that Phlox missoulensis comes in three different color morphs. So hopefully you can see this okay on your screens. But there's sort of the most common one is a very bright white. But then they can range in various shades of blue. And then there's also a pink morph that takes on this kind of reddish hue. And so last summer, we set out to just sample the trait variation across the known populations that we had access to. So we collected about 100 floral samples from 8 different sites that spanned 2 preserves, so Waterworks Preserve as well as Bluebird Preserve up by Grant Creek, if you're from Missoula. And those 8 sites at those 2 preserves, we roughly grouped as 3 different habitat types. So the background of these communities could either be pretty grassy, they could be very much like gravel soils, and then at Bluebird, the phlox is actually found. You can kind of see the phloxes kind of up front here is found in these pits that are left over from the quarry that's right behind Bluebird. And so it's gravel soil, but it's reddish. And so we think that there's some maybe heavy metal accumulation in there or some other type of pollution that makes this habitat a little bit different than the gravel habitats. And then obviously they also have a very different history of disturbance because it's, just quarry kind of dumping in the back of the preserve. And so what we did was to get these 100 samples, we went out and we took floral pressings. So we just pressed flowers flat onto Scotch tape onto a sheet where we can have a size standard as well as color standards. And I like this image because it shows our phlox individuals, and you can see everything from very, very white flowers with pretty narrow petals. We have some of our pink morphs on plant 1012, very purple in 1004. And then we can also see the variation in these fat petals in, for example, plant 1015. And so a ton of variation, and this is just from one of our sites at Bluebird. And so what we did was we took these floral pressings, and this is work led by Julie Cox and Max Hirl at UM. Um, and they used imaging software to be able to extract floral color as just red, green, blue RGB values. So here's again a smattering of the floral pressings that range that variation. They were able to extract floral diameter, so how wide the full flower is, petal width and lengths to get at, long and narrow versus short and fat petals. They also measured corolla tube length, so this part kind of under the petals down to where the ovary would be found, so this red line here. And then they measured leaf length and width as well as how thick the leaf is, which is a trait called specific leaf area. And so we found a couple results that did differ with habitat types. So what I'm going to be showing you here is a different trait on this y-axis. So in this case, I'm showing you the red coloration, so the R values from an RGB extraction. And then the 3 habitat types, grass, gravel, or pit on the X. And so what we found here was that the pit population here has somewhat lower amounts of red pigment in it. So the flowers tend to be a little bit more white is what that's telling us in the pits. We also found in the pits that they have what I would call a somewhat elongated growth form. And so here I'm showing you 3 traits. So I'm showing you floral diameter in this leftmost figure. And so here we can see that in the pits, the population average, so this pink diamond here, is a little higher. And so that tells us that the diameter of the flowers is a little bit stretched out in the pits. And just so you know, so these plots are kind of funky, but all these dots are the raw data. And so this is showing that there's a lot of variation in floral diameter just kind of across the board. And then this shape, this is a violin, is showing you kind of the bounds of the spread of that data. So it's just showing how variable things are. We also found that in the pits, they had a higher value of specific leaf area. And so for a higher value of specific leaf area means a thinner leaf. So we see this value here. And we also found that corolla tubes were a little bit longer in the pit, so this higher value of corolla tube length. And so this suggests that floral diameter, leaves, and the corolla tubes are all just a little bit stretched out. And so we were a bit confused by this, but we did find some evidence in previous studies in a gladiola that metals like iron, copper, and zinc can lead to these kind of stretched out floral characters. So one of the things we're really planning to do is we've got soil samples from all of these sites. We're going to be sending them off for sampling soon so we can get soil nutrient as well as heavy metal data in these sites and actually ask, do the pits have more iron, copper, and zinc that could have led to this pattern. The other thing to note is that thinner leaves, so this middle plot about specific leaf area, thinner leaves usually correlate with more stressful conditions. And the pits, they're very exposed. They have this history of quarrying. They have this potential heavy metal content. And so we think that could be, contributing to that trait as well. The last thing I just wanted to show you, we measured a whole bunch of other traits. So things like the blue coloration, um, everything was exactly the same amount of blue, so not an interesting result. Petal length, we're again seeing a trend for longer petals in the pits, as well as longer leaves in the pit. So all kind of pointing towards that elongated growth form. But what I want you to take away is that all of these traits show a ton of variation. So the species is a little bit of a mess in terms of being able to delineate, these are the traits that would make it a Missoulensis versus something else. And one of the things that my lab is super interested in moving forward is digging into this color variation, because it's really like these are three very stark color morphs. And we have two key hypotheses for why this happens. So these floral color dimorphisms, so where an individual species can come in at least two different colors, can occur via soil conditions. And so some examples of this are Leptosiphon parviflorus. This is a California species. The pink morph right here is found on really harsh serpentine soils. And the unpigmented white morph is found immediately adjacent to it, but on sandstone soils. So the soil condition basically controls the color of this plant, even on a very, very small spatial scale. Similarly, in Linanthus, there's a blue and a white morph, and these blue and white morphs kind of have two things going on with them. So the first is that they grow on two sides of a ravine. The west side of the ravine has the blue flowers, and it's a side of the ravine that's very wet and has a bunch of other species, so high species diversity. The east side of the ravine is a lot drier. It has the white flowers, and it's more dominated by creosote bush. And so there's this spatial variation, and then where the populations are mixed, which is a image that I'm showing you on this slide, years with higher soil moisture favor the blue morph. So that morph that is on the west side in that wetter microhabitat. The other thing that can drive these floral color dimorphisms is pollinator choice. And this is where pollinator perception, kind of zooming around, looking for different flowers, affects affects the floral traits because they're picking who to actually pollinate. So one sort of classic example of this is the evolution of UV signals. So pollinators are going to select for these species that have these very distinct UV patterns, usually in the center, that point them directly at the pollen and the nectar that they need to get to. The second way that this can happen, which is a little more roundabout, is via floral background. And so in these cases, we have some plant, in this case, that has blue and purple wavelengths. They're growing in some light environment that's affecting how well the pollinators can see. But there's also some background context. And so you can have backgrounds that are very sort of brown or that are kind of green and maybe harder to see through. The pollinators are going to be able to see different wavelengths. And so the idea here is that if you have the same plants growing on a dirt background, a brown background versus a green background in grass, theory basically predicts that in the brown backgrounds, darker flowers might evolve, and in the greener backgrounds, lighter flower might evolve. And that lighter flowers is so that a pollinator zooming above could be like, oh, look, I can see this giant white flower. Let me go pollinate it. And so a really nice example of this is in Appalachian Phacelia. And so in this species, they grow on rocky granite outcrops in the Appalachian Mountains. And on outcrops, they tend to be this purple morph. And if they're growing in the grassy habitat around the outcrops, they tend to be this white morph. So this lighter, bright white on a grassy background. So moving forward, we're going to be thinking a lot about this floral polymorphism, so three color morphs in Phlox missoulaensis. What we know already is that the pits have less red flowers. We also know that 2025, which was a drier year, had way less color variation than 2024. Um, so 2024, we saw all these blue and these pinks, and last year we didn't really see that too much. We have a bunch of other data on floral color, and then as I mentioned, we have some soil cores from each site to really think about heavy metals and soil nutrients. We also have local data on things like soil temperature and moisture, so we're going to be able to test whether this color variation is driven by something about the soil. So the first things we want to do is just say, at the site level, do these soil conditions matter for average floral color? And then does it differ between the grass and the gravel? So the green, a green versus a gray background. Next summer, we're also hoping to do that at the individual plant level. So go out to an individual plant. Let me go back. So an individual plant, get an image of its color, but then also stick, some soil monitoring equipment right next to that plant, get some information on the soil conditions, and then also get some measure of the background color. And we can do that kind of by using this color standard. And then in this image here, the pink circles are circling flowers. The yellow circles circle like a spot in the background, and we can look at the contrast between them. And so hopefully that will lead us to some other cool experiments, looking at, yeah, whether there's some adaptation to soil conditions. We can also look at whether pollination is better for certain color morphs on different backgrounds. So, for example, we might expect, based on the Phacelia, that there's going to be a lot of pollination of a white flower on a green background because it's highly visible to pollinators. And maybe that's less true on a gray background. Okay, so as we've been talking a lot about pollinators, we've also been kind of picking at some of the reproductive biology of this species. It's kind of shocking how little we know about even how much it reproduces. So the first thing we've been trying to figure out is just whether it's self-compatible. And so this past summer, we tested for autogamous selfing. So this is a form of selfing where the stigma on one flower will kind of bend back and then it'll pollinate that same flower. And this is a good strategy if you think you're not going to get pollinated because then you're at least giving yourself some pollination, but it means that you're getting the same genes from the same flower. And so it means that your offspring are going to be really closely related to you and so really inbred. So this past summer, we bagged 3 flowers on each of 25 plants. Not a single one produced any fruit, so we can pretty definitively say no autogamous selfing is happening in for this system. The other thing that can happen is that species can be geitonogous. And so in that case, especially for species with really large floral displays like these guys, a pollinator might visit one flower on the plant, but then go immediately pollinate a flower on the same plant. And so that's, not the same as the stigma kind of doing it itself. It's still pollinator driven, but you're still getting really high levels of inbreeding because you're getting genes from your same individual. So for this, we've collected seeds from open flowers on those 25 plants. And what we're going to do is we're going to germinate them, get tissue from the offspring. We can then extract DNA and say, is the DNA from your mom like the same as your other parent. In that case, they're selfed, whereas if the DNA from the mom is very different from the rest, we can say, okay, your paternal plant came from somewhere else and you're outcrossing. So we'll hopefully figure that out in the next year or so. The other things we know about reproduction is that the color morphs do vary in their compatibility. So a 1992 master's thesis did a pilot study on this, so we'd love to redo it. And they found that the pink and the white morphs freely interbreeding. They, produce plenty of seeds when they get crossed. But the blue morph is off doing its own thing, so it basically only crosses amongst itself. And so don't know what's up with the blue morph at this point. And then we also know that these guys generally produce about 3 seeds per fruit. That is actually pretty conserved across the entire Phlox genus. It's generally about 3. So the other thing within reproductive biology that we've been focusing on is pollination. So we've been doing some pollinator observations, as well as thinking about how the neighborhood of who's flowering around you facilitates pollination. And so, as I mentioned at the start of this talk, Missoula phlox is embedded in this matrix of other cushion flowers, including the primrose, the cushion buckwheat, the erigeron, the cutleaf daisy, as well as a ground cell, Pachyrhacana. And these guys, so this is showing you the names of the species on the y-axis and then just dates on the x. And so phlox is up here in pink. And so this is saying that phlox starts flowering in 2024. It started flowering, I don't know, like May 3rd on average. And then it flowered through. So here's May 3rd here. And it stopped flowering, I don't know, May 20-ish, somewhere in there. And so what we can see is that the phlox flowering overlaps a little bit with the Douglasia montana, DM in green. It overlaps almost entirely with this teal species, which is the erigeron, the daisy. The tail end of it overlaps a little bit with the buckwheat. And then this is the Pachara, and in 2024 it didn't overlap, but in 2025 it did. So we use the Pachara as a pair as well. The point here is that Missoula phlox is therefore competing for pollinators with these other species that it co-flowers with. And so these floral displays in a patch can either really help pollination, so be facilitative, or can hurt pollination if it's causing competition. And so theory, again, suggests that pollinators, if you're zooming around looking for flower patches, you're probably going to visit patches with pretty high floral density. So that's called a concentration effects, where as floral density increases on the x-axis, the per flower visitation rate of the insects should increase until they've kind of gotten the visits that they need, they've gotten the pollen and the nectar that they need, and so that's going to level out at some point. But there's also that they might visit proportionally fewer flowers when density increases. So a bit of a dilution effect where if you have a ton of neighbors right around you, you as an individual plant are going to be less likely to get a single visit. And so there's evidence that these floral displays, the effects of them can be really scale dependent. So this is a study in Trifolium pratense, red clover. And in this situation, the researchers looked at other purple flowers that could be attracting bumblebees at 2 different scales. So at the park scale, so a 10 to 25 meter radius around some focal clover plants, or at a very local scale, so immediately around the focal plants, only within like 2.5 meters of their patches. And what they found was that at the big park scale, pollination visitation increases the more purple flowers you have to attract the pollinators. But at a smaller spatial scale, you don't want to have a ton of neighbors. So if you have a lot of neighbors over on the high end of this axis here, your pollination rate starts to decrease pretty quickly. So a new master's student in my lab, Keely, basically set up this experiment this past summer where she paired Phlox, which is this species here, with one of the 4 species that was that it co-flowers with, so an immediate neighbor that it would be competing for pollination services with. She then also looked at sort of the immediate neighborhood around them, so how many flowers were open within this close neighborhood within only zero point 0.5 meters around it, and then a farther neighborhood at the 3 by 3 meter scale. And so she took these 10-minute video recordings, just watching for pollinators on 197 different focal phlox plants, and then photographed and counted the number of flowers in this close neighborhood in green and this blue neighborhood, or this far neighborhood in blue, sorry. Um, and I'm just going to tell you now, the effect of immediate neighbor didn't matter at all. So whether you were paired with a primrose, a daisy, a buckwheat, that did not affect pollination. So I'm going to show you the results for the close and the far neighborhoods instead. And so what we thought would happen would be that in your far neighborhood, you would want more flowers, such that more flowers, I'm on the wrong screen here, more flowers would lead to more visitation because you're a pollinator and you're attracted to these patches with a ton of floral diversity. Whereas in your close neighborhood, if you had a ton of flowers right around you, the pollinator would be more likely to go to your neighbors than to you yourself. And so the phlox might suffer from the fact that immediately around them, there'd be a lot less flowers. So in her recordings of pollinators, so we looked at just who was visiting first. So the biggest pollinator group, so this is number of visits as a function of these different morphotypes of pollinators. Morphotype 1 is Bombylis, which is bee flies, vastly the largest pollinator, daytime pollinator of the phloxes. Some visits from bombus, so from bumblebees, as well as lystris, which are flower beetles. A handful from morphotype 4, which is Andrena, which is a really large group of solitary bees. And then some visits from thrips. The thrips are not probably pollinating. They basically suck tissue out of plants, so they're probably hurting more than anything else. And then a couple visits also by Apis mellifera, which is honeybees. Huge caveat to this. So these species, as you saw, have these really long corolla tubes. So it's a very high probability that they're getting some moth pollination. So one of the things we'll be doing this coming summer is some night surveys to actually look for moths, which should be fun. Okay, so what Keeley found, so this is showing whether or not a plant was visited on the y-axis. So visited, yes, no. And then this line is showing you the probability of visitation. So when it's a little bit higher, that means an individual phlox had a high probability of being visited and pollinated. And then when it's low, lower probability of visitation. And so we saw that these neighborhood scales mattered for pollinator visitation, but not in the way we expected at all. So we expected that in the far neighborhood, you'd want to be in a huge patch of flowers so that visitation would increase. So that's up in this corner here, this orange line. And what we found instead was in the far neighborhood, the exact opposite. You wanted very few flowers around you for pollination to occur. For a close neighborhood, we expected that you would get more visitation if you had fewer flowers immediately around you, such that pollinators would, potentially go visit your neighbors. And instead, we found that the more flowers that were in that 0.5 by 0.5 meter quadrat, that very small spatial scale, the probability of visitation went way up. And so what this tells us is that this might be, rather than about attraction via floral displays, it might be about pollinator foraging patterns. And so in a far neighborhood, so this big spatial scale, pollinators have a lot of options about where to start foraging. So this is a picture of our site at Bluebird. All of this white stuff is all flowering phlox. Focal plants are tagged with these pink flags. There's a lot of options about where to start foraging, and so any single plant is less likely to be visited. But then once a pollinator chooses an area to forage in, they might be more likely to stay and forage in that very small area. If that requires less travel, maybe takes up more energy, they can just pop from flower to flower to flower without really flying. And one way to visualize this is two quadrats where this is the focal phlox species. This plant has not a lot of neighbors at all. And so a pollinator would kind of finish up and then have to go somewhere else. Whereas in this transect, here's the focal phlox right here-ish, kind of extends over to here. But there's a lot of, in this case, phlox and the daisy around it. And so a pollinator could just pop from flower to flower to flower for quite a while. So it might be less energy, it might be more energy efficient to just kind of hang out in that small spatial area for a while. And we think this has some implications for where to outplant or seed Missoula phlox. You're going to want to be in these smaller patches where, a pollinator doesn't have a ton of options about where to go, but in these very dense, small areas where there are a bunch of flowers in a very concentrated amount of space. Okay, as we move from reproduction biology, we spent some time on sort of the flowers and the seed, like the pollination and what would affect fitness. I want to move into talking a little bit about some of our population monitoring and sort of trends of population health. So for our population monitoring, we've been monitoring 6 cushion plant species, including the phlox at Bluebird and Waterworks preserves. In each of these preserves, we have 3 subsites that vary in the grass versus gravel habitat type that I talked about earlier. And then within each of those, we have a transect, so a straight line marked with 50 plants marked, at least 50 plants marked. And so what this looks like is we can lay out a tape. Every plant has a coordinate along that tape. And then it's also tagged with a metal, aluminum metal tag, so that we can go back and find the plant year after year. So if you've been to like the top of Waterworks and you see a bunch of really shiny stuff, that is our lab. And so every year we go out and we measure survival from one year to the next, the size of the plants, um, reproduction. We get an estimate of seed production, and then soon we'll start monitoring germination as well. But we can monitor each of these different vital rates, survival, growth, reproduction, over many years, and then put that into these models that basically predict population growth rates. So whether the population is growing or declining over time. And so, huge shout out to my lab here. This is a huge effort by a lot of people every year. Also, if you see huge crowds of people bunched around like 2 plants at Waterworks, that's also typically us. But particularly, Minody and Dalton have really taken the lead on this population monitoring. Okay, so this first figure I'm showing you is for survival. So this is showing you survival between the 24 and 2025 field seasons. I'm showing you the results for every site on the x-axis, but all of the Bluebird sites are in blue, and all of the Waterworks sites are in these different shades of pink. And so what I want you to note is that survival is really high across the board. So even the lowest survival rate is 97.5%. Um, and then there's actually absolutely no death at Bluebird, at least between the 1 year, the 24 to 25 season we've been monitoring. So this is great. Survival is really, really high. Similarly, so we see that reproduction, so this is the average number of fruits that are produced on any given plant across the sites. Um, it's actually quite a bit lower at Waterworks, and then there's high reproduction, relatively speaking, at Bluebird. And then finally for growth, this one's a little funky. So the way we measure this is we get the area of the cushion. So we measure the 2 axes of the cushion plant, and then we basically make this ellipse and estimate the area of the ellipse. And so what I'm going to be showing you here is log of the area, so that ellipse shape in the previous year, so in 2024 in this case. And then on the y-axis is going to be log of the area this year, so in 2025 in this case. And so if plants are not growing at all, they should fall along this dashed 1-to-1 line where log of the area in 2024 is exactly equal to the area in 2025. So any plants that are above this line are plants that are growing, so getting bigger, contributing to the population. Any plants below this line are going to be shrinking for some reason. And so this is kind of a crazy looking graph, but what I want you to note is that so these smaller plants, so kind of the lower end of this area, are all getting bigger. They're mostly above this dashed 1-to-1 line. So small plants are getting bigger. What's interesting here is that once you kind of get to these larger plants, so the huge cushions that can be the size of like one or two watermelons, they drop below the 1-to-1 line. And so these large plants are beginning to shrink. And I realize this is hard to see here, but more of these shrinkages are in the pink dots. So it's worse at waterworks than it is at Bluebird. And so if we go back to this kind of life cycle diagram, what we're seeing is that survival is really high, especially at Bluebird, absolutely no death. We're seeing that reproduction is the highest at Bluebird. And we're seeing that large plants are shrinking, but especially at Waterworks Hill. Oh, and then I'll just point out we haven't done germination yet, but we'll be setting up plots to start that in 2026. So what this indicates to us is that based on survival, growth, and reproduction, Bluebird likely has the most stable populations. And Bluebird Preserve, has its own history of disturbance, but it also has the lowest foot traffic. So this is data from Parks and Rec, from these counters that they put out this past spring. Bluebird is in blue, unsurprisingly. This is the number of visitors just over time. And then Waterworks is in green, and you can ignore the black. And so Bluebird has way less foot traffic. Um, and what I also want to point out is that more shrinkage at Waterworks, which is the high traffic site, has these really large plants that get stepped on because the trail runs kind of right through their habitat. Whereas in Bluebird, the sites are a little bit away from the trail, so they're a bit more protected. So we're trying to more directly link some of those foot traffic impacts to the Phlox population growth rates, and we're really interested in setting up some monitoring and some other sites that we know about that have virtually no foot traffic kind of as a control. Okay, and then the last story I want to tell you guys about is focused on invasive species. So I'm sure I'm talking to the choir for this group, but invasive species can significantly influence where and how much we find, so the distribution and abundance of different native plants. On Waterworks Hill and at Bluebird, kind of the big threats are going to be cheatgrass, leafy spurge, and Dalmatian toadflax. And these invasive species can benefit quite a bit from disturbances like trails through their habitat. And so what we expect to see is that as you move farther from the trail, we would hope that the abundance and the richness, so the number of different species of invasive plants in green, would decrease, hopefully pretty rapidly. So a lot of invasives right near the trail, right where things are disturbed, and then decreasing from there. And then we would expect that where the invasive species, oh, I lost my cursor. Where the invasive species are low, we would hope that cushion plants, could kind of establish in that space and do pretty well. And so we'd expect that cushion plants would be a little farther from the trail. So an undergrad in the lab, Audrey, also last year decided to ask how distance from the trail affected the native cushions versus invasive species all the way along Waterworks Trail. And so she did a massive amount of work. She measured diversity, so species richness, the number of unique species, as well as abundance, so just a count of the individuals across 360 quadrats, all from the Waterworks Trail, kind of from where there's a big radio tower all the way up to the summit, for those of you who are familiar. And she wanted to look at the differences in the diversity of the invasives versus the cushions as a function of both distance from the trail, but also side of the trail, east versus west. And so I'm going to show you some results showing these diversity metrics as a function of these distance from the trail. So these high values here will mean that it's farther from the trail. I'm going to show you this split between the west side of the trail and the east side of the trail. So the west side of the trail is the side of the trail that's facing Missoula. So if you're walking up towards sort of the overlook area, Missoula is going to be on your left. This part of the trail, it's facing Missoula. The ground is fairly flat. On the east side of the trail, this is the trail facing Cherry Gulch. It's very, very sloped. And so there's this ridge line, and then there's a slope where we would expect more runoff, potentially wetter soils. And it transitions into this more grassland the farther down slope from the ridge that you get. And so what Audrey found, so this is the number of unique cushion plant species in pink, number of different invasive species in green. On the west side, we find that species richness increases with distance from the trail. So this would be is what we would expect when there's more disturbance, so very close to the trail, so the left side of these graphs, there should be fewer species. What's really interesting is the east side. So on the east side, as I said, the terrain is more sloped, potentially more runoff and soil moisture. And so what we're seeing is that close to the trail on the ridge part is where all of the cushion plants are. So they're in this disturbed area, but they're on the part of the trail that's very dry. And then they drop off the farther down slope you go. And that trades off directly with the invasives. So the invasives, completely opposite of what we expected to be happening, are not very common close to the trail and increase dramatically farther from the trail. And in this image back down here, you can basically see the farther you are, so the trail's like right here in the bottom right corner of this photo. You go down slope and you're basically walking into a cheatgrass field. And so that's what's happening there. So the cushion plants and the invasive species are really occupying these super distinct areas of waterworks. And if we look at abundances, so this is just count of the number of individuals of each of these two groups. Again, cushion plants in pink, invasives in green. On the west, we're seeing more invasive species over here, close to the trail. So exactly what we'd expect if disturbance was favoring the invasives. But on the east side, we again see that the cushion plant abundance is really high, very close to the trail on that ridge line and decreases, and that trades off with the invasive species. So again, there's this really sort of distinctive spatial separation going on. And so the next step for this is going to be led by a master's student in the lab, Keely. She's going to be doing some species distribution modeling, trying to ask if things like soil moisture, wind speed on these ridges, presence of the invasives, is that limiting the native species from moving down the slope or vice versa? What about this habitat on the ridge is preventing the invasives from just moving up slope and completely outcompeting all of the cushions? And we'll probably expand that to some cushion plant sites across the Missoula Valley to think about how moisture and winds, invasives, and then trees, which would block some of the winds, affect native plant distributions to help hopefully identify some sites to protect this really kind of vulnerable set of species. Okay, so tonight I've told you kind of a bunch of different hodgepodge stories as we've been picking at the system in these 5 different themes. And I think what we've learned so far is that for taxonomy, we've identified a bunch of potential sister candidate species. We really need to get into the gene flow between these populations and the relationships between the species to get a better sense of how the species, came to be so spatially and temporally isolated. We've also found that there's a lot of trait variation. There's a little bit of a pattern going on where the pits have this weird elongated growth form. And we're super interested in the color variation, but we're still learning a lot in that theme. We've found in terms of reproduction that there's pretty limited self-compatibility, that this species is very much pollinated by generalist pollinators, and that it needs really close neighbors to get pollination to occur. So we want to plant in kind of these dense patches of neighbors. In terms of population trends, we're seeing that phlox performs best where there's less foot traffic. That points to some nice potential land sort of management decisions that could be implemented. And then finally, we're seeing that cushions occupy a really distinct space from invasives, which leads to the question of what are some of these filters, so moisture, wind, et cetera, that restrict their distributions. And so to wrap up, I think this species is really interesting and worth protecting in its own right because it is a Montana endemic species. That's not something that a lot of places can say that they have. And I think it really gives a value and sense of place to a lot of these preserves. Um, but it's also this member of a group of really taxonomically challenging phlox species that are distributed across the mountains of Western North America that encompass both a lot of trait variation and a lot of habitat variation. They span all these different habitats that could be doing quite different things. We're seeing an increasing amount of evidence that there are threats from foot traffic. So hopefully one of our next steps will be working with Missoula Parks and Rec to help identify some potential management interventions to protect the current habitat where it's at. Um, and then really broadening back out from a science perspective, if this endemic species is in the process of speciating, becoming its own thing from one of these more widespread species, we can really use it as this model system for why things become endemic. And that could be really useful for thinking about sort of endemism more broadly across the globe. All right, and with that, I would love my lab has contributed so much to this work. So the folks who are asterisks are taking have taken lead roles on some of the projects that we've done. Also, huge shout out to Missoula Parks and Rec for not only giving us access to these sites, but talking to us about like all of these experiments and all of these ideas that we've had. Funding sources largely through the Northwest Climate Adaptation Science Center at UW, University of Washington, as well as UM. And thank you all for listening. And yeah, hopefully we can spend a good chunk of time on questions.
Thank you, Meredith. Um,
I can stop sharing. That might,
oh,
yeah.
sure. Yeah. And sometime, yeah, if you want to keep sharing, sometimes
Oh,
we
if I need to jump back
scroll
to something,
back, but yeah, no big deal. You can,
I can do that.
so I'm just gonna scroll back, to, I think what I'm seeing as the first question that we had, which was, and I think you ended up addressing this, but it's always nice to have a repeat.
Yeah.
So Kathy Sedevendemi asked, do individual plants change floral color with annual conditions, or do they remain their original color? Does this explain changes in color within populations where you have one year more white than others? So yeah, maybe that is a pretty fresh question.
It is a pretty fresh question. Honestly, we don't know. So we haven't like tagged the same individuals and then monitored their flowering time year after year after year. My gut feeling is that they must change color because of exactly what you said in your question. The fact that like the pink morphs that we saw in 2024 were obviously not that pink in 2025. I'd want to confirm that it wasn't that those plants just like didn't flower the next year or something like that before saying like definitively that that was the case. Um, but it does seem like it's a pretty flexible trait that my guess, I think it's more likely to be the soil conditions, so that soil moisture factor, and then it does change year to year.
Yeah,
But
I'm
to be
so excited.
determined.
I'm excited to hear the results of the soil tests.
Yeah, me too.
Keep us posted. Maybe your talk in March, maybe you'll have those.
Maybe, maybe.
Okay.
It's
For
a good goal.
Missoula locals, we'll see. Um, okay, great. And then Connie Geiger asked, given the impact of foot traffic, how do you factor in your own impact when studying and measuring your transects?
Yeah, this is a perennial problem with ecological studies. What we do is we try to dance around the plants as much as possible. So you can kind this image actually works pretty well. There are these patches of soil. We try really hard to step mostly in those so that we're not stepping on the large plants. But that's kind of as good as we can get it for now.
Yeah. I'm so I Waterworks is one of my favorite places to walk, and I really am so impressed with like the effort that Missoula Parks and Rec has done in sign, like
Yeah.
really being aggressive about the signage. Like with dogs on leash and just the awareness building.
Yeah. And I think they've hit a good like happy medium of, putting the signage up more when the phlox is flowering and super
Yeah,
vulnerable. Because if the flowers get trampled, then they can't produce fruit. Um, and so that's like, gives kind of the little bit of an option of, okay, we want to have our dogs maybe sometimes being able to be off leash. So let's avoid at least the sensitive stage of the plant kind of deal.
yeah.
Um, so a nice intermediate there.
And then Connie also asked, since their traits vary a lot, do you have any tips for identifying the species compared to their lookalikes, or would it require keying it out?
It does tend to require keying it out. Like when we've gone out to these field sites to try to collect tissue from the other populations, menodes tend to have to key them out. Roughly, I would say if you're seeing a species with kind of shorter leaves that looks like the leaves are really thick and shiny and kind of like a succulent plant almost, you're probably looking at Kelsey. Um, and then if you're in a higher alpine area and the cushion is like very distinctive, that's probably pulvinata. If you're in the Missoula Valley, it's basically it's going to be Phlox misulensis. We don't have much of the Kelsey and certainly not the pulvinata in the valley itself. So space helps a lot for the IDing.
Right, because they're so particularly located.
Yeah. Yeah.
Vicky Watson is asking, would very careful weeding by knowledgeable citizen scientists be helpful on Waterworks Hill?
I'm gonna say yes. I have definitely like when people talk to me, especially at Bluebird, and they ask what they can pull, I tell them to pull the Dalmatian flax or
Toadflax,
Dalmatian
yeah,
toad flax,
yeah.
kind of as they're walking. So yes, but it would have to be very careful, and you'd have to be very very comfortable with your plant ID. But at Bluebird, I definitely have told people that in passing.
And if I'm remembering correctly, the Missoula Parks Department has done at least one weed pulling.
Yeah.
So get in touch, check, take a, especially Vicky, if you have excited students, the Parks Department, I know they organized at least one weed pull. So that's always a good way to do it. Weeding community.
Yep. Then you
Uh,
know what you're pulling, and yeah.
yeah, yeah. Keela Murdoch just said that they wanted to give their gratitude, just a nice note in there.
Oh, thank you.
Um, Rachel and Jack Potter asked, has there been any monitoring done in the past?
Yes. So Peter Lessica in the last couple of years has done some frequency surveys, particularly at Waterworks and Mount Jumbo in different areas of the phlox, plus a handful of the other cushion plant species that we're now monitoring. So it's a slightly different way of getting at population abundance. But like his data has pointed out that, the phlox populations are pretty stable. Um, and then the primrose populations and the buckwheat, I believe, are the two that he's seen some declines in at these sites in the last few years. So yes, there's been some monitoring. We're doing it in a subtly different way. But yeah.
And oh, I'm trying to say people's names to give them some credit, but that means I'm probably going to pronounce them wrong. Nakaya is asking, are you testing the pollen quality on plants?
We are not. Um, we could try to do it for pollen quality because these plants, the phlox in particular, produce a lot of pollen. And so understanding like whether it's very nutritious for things like the bees would be super interesting. What we did try to do last summer was get nectar to try to get nectar quantity and quality. Um, and we got almost no nectar out of these plants, so I don't think they produce very much. But yeah, we haven't done pollen yet. That's a great idea.
Interesting. Very A lot of praise in here in the chat. So yeah, lots of people really enjoyed this presentation. Andrea Pip is saying, I think it's great you are looking at Missoula phlox and the cushion plant community with a variety of questions and studies touching on genetics, pollination, morphology, taxonomy, and plant to plant competition. It's uplifting to hear a lot of interest in a local plant community. Thank you and your students and collaborators at Missoula City Land Trusts, and others. That's sweet.
Thanks, Andrea. You've helped with
Um,
this too, so.
oh, I'm sorry, Andrea. Andrea.
Or
Um,
I
oh, okay. um, let's see. James Habeck, sorry. Um, just added that back in 1960, back in the 1960s, Missoula Valley was impacted by constant toxic air pollution coming in from the Frenchtown paper mill. People and plants were harmed, and Jim was a plant ecology professor at the from 1960 to 1995. So it sounds like
Oh,
Jim, you maybe have some insights to things you saw. Maybe we'll have to be in touch. Cassandra was asking, is seed banking in the works?
it will be eventually. So in terms of a seed bank for these guys, we will need to test it. Alpine species, normally we kind of assume just based on a lot of them not having a seed bank that they don't need it, but I don't think we can make that assumption in this system because it is lower elevation. Um, so seed banks in the sense of like seed bank in the soil. At some point we basically bury nylon mesh bags of seed with a known amount of seed, and then we can quantify how much of that within that area germinates 1 year post burial and 2 years post burial, and that tells us something about the seed bank in the soil. And then if you're talking about seed banking as in saving seeds, yes, we're collecting seed every year. Some of it is going to get used for some experiments this coming spring, and then some of it we'll kind of stash. So depending on that answers both versions of seed banking. So hopefully it gets at the question.
Awesome. Um, Keela, I hope I'm saying your name right. And I'm sorry, I think I saw this question way back in the past, but thanks for mentioning it again. Out they're saying, out of curiosity, what are the root systems like?
The root systems for cushion plants are kind of neat. So they tend, if you pull up a cushion plant, they look like little trees. So they're super branchy at the top, and then there's a tap root typically.
Mhm.
Um, yeah, they don't go like crazy deep, like nothing like a prairie plant, but enough that they, are accessing below ground resources.
Very cool. I think that's all of our questions in the chat. I want to just shamelessly plug that if you are not able to get onto Waterworks Hill or these other locations, you can in fact see Missoula Phlox along a paved pathway at Rocky Mountain Gardens in Missoula. That's where I work. But in our crevice garden, we have Missoula phlox growing. Meredith will be there in April talking about it while it's growing and hopefully blooming.
Hopefully.
I hope. um, but yeah, so if anyone has a question, like yeah, the Rocky Mountain Gardens, we do have a few specimens growing. So if you want to see it up close and personal, and the trail is not an option for you, please come visit us. And let's yeah open the floor for other questions. If you just want to unmute yourself, we can try it that way.
I'll also pitch while people are thinking that there'll be a herbarium night this year. One of them, the one in the very end of March, will be Missoula cushion plant, so phlox plus some of the other species that I mentioned. So
Very
it's not
cool.
up and close and personal with a live plant, but it's up and close and personal with a dead plant.
Any questions from the audience? Kelly, thanks for adding that. Hi, Kelly. Yeah, Kelly mentioned that they also have the Phlox and the native gardens,
True.
the Montana Native Botanic Gardens on campus, which I had the pleasure of visiting so recently. So, yeah, yeah, another awesome accessible location.
I
Oh,
also see a
and
question, can
yeah.
this grow in a home garden now that I'm not sharing screen? Um, Maybe, but I think it'd be pretty hard for a couple reasons. So you're going to need like very, very rocky, dry soil, which could be pretty easy in Montana. So I think it could. The other thing is that the seed that's available via like local nurseries, and Alex, you might have a better answer for this, is pretty limited because you don't want to collect a ton of this now S1 listed species. But I think you told me there were some groups that were still trying to like propagate it in nurseries for sale.
Yeah,
Yes.
oh, it sounds like someone else has it looks like Kathy said of Endemi is growing it in
Growing in a native
her native
garden.
garden. Oh, yeah, I wonder if that's true. We'll have to ask Elliot that. Kelly's saying Elliot at Pipolo might have it. We'll have to ask when he's presenting. Really excited for that talk for people who have cabin fever at the end of March. But yeah, I know Molly, my coworker Molly at the Gardens, when she worked for the city, that's when she was able to collect seed, and that's the seed that we have in at Rocky Mountain Gardens. Kelly, I'm wondering if you know where the Missoula phlox in the Botanic Garden at the university, if you know where that came from.
Um, yeah, I think it was collected by Klaus Lockchevitz. So
Okay.
it's old. It's probably
Oh,
a 50-year-old plant.
wow.
It's declining a little bit, but that was the digging time. They dug it but it's lived and stayed in a good draining, sunny, hot area.
Any other questions? Yeah, feel free to enter into the chat or just unmute if you want to speak up. Meredith, so it sounds like there might be a couple other opportunities for people to learn more about Missoula Phlox in this or this
This
spring.
spring. Mm-hmm.
Okay.
Yeah, so I'll be doing a Blooms and Brews talk in February,
March?
maybe
Yeah.
March sometime. And that will be, I mean, some of this again, but hopefully we'll have some of like the soil and the climate data worked out. And then, yeah, we'll have one of the themed herbarium nights at the University of Montana Herbarium this year will be cushion plants. But there'll also be herbarium nights on tropical plants and botanical illustration and astragalus and at least one other. So,
Very cool.
yeah, be on the lookout for those announcements too. I think that'll be through the local Native Plant Society chapter.
Hardcore chapter. Okay. Well, if there's no other pressing questions, thank you everyone for joining us. I believe we hit 106 people, so that's really remarkable.
Oh,
Um, very, very awesome. Please come back to join us in December. You will be getting updates. You can visit the Montana Native Plant Society website to check on the schedule, put it in your calendar, set an alarm. And as always, if you ever have an idea for a talk like this, while this season is full up, I'm always stashing new ideas away. So let me know. It would be wonderful to keep the pool of options open. So thank you, everyone, and a special thanks to Beth Madden, who has helped me manage this duty in her wake. So really grateful to have her on board to help me through this. But thank you so much, Meredith, and we will see you all again soon. Take care.